Hello, everybody. My topic is DNA and its structure during different types of processes it goes through. So, what is DNA? It is a deoxyribonucleic acid. There are four main bases, shortened as ACGT. A is adenine, C is cytosine, G is guanine, and T is thymine. So you might wonder, which bases pair up with which? DNA pairs up as adenine with thymine, cytosine, and guanine bases. Each of these bases attach to a sugar molecule and a phosphate molecule. So the ladder structure formula is a base plus a sugar molecule plus a phosphate molecule equals this equals a nucleotide. And when a nucleotide is in two long strands, a double helix is formed, which is the basic ladder structure for DNA. As we continue through this lesson, you might be asking yourself, who discovered DNA? The story of the DNA discovery seems to start in the year 1928 with scientist Frederick Griffith, whose experiments with mice bacteria helped him learn how genes can transmit disease to offspring. However, less well known that Frederick Meister in 1869 chose leukocytes as his source material in investigating the proteins in these cells. He found a different discovery, which was when he noticed a substance with unexpected properties that did not match those of proteins found in past experiments. And he didn't know, but he actually found the first true purification of DNA. And where is the DNA located? It's in the cell's nucleus. And what does DNA do for us? The purpose? Used for organisms to survive. There are two types of processes I'm going to discuss. Process number 1, replication. An easy way to remember it? It just makes copies of itself. Each strand in the double helix can serve as a process to duplicate the sequence of bases. A DNA sequence is also known as a gene. The way it does that, they unwind so they can make multiple copies of the different strands. Process number 2, DNA into RNA. So there are 3 steps into remembering this process. The first one, enzymes are the researchers and read the information from the DNA molecule and transcribes it into a molecule messenger called ribonucleic acid, or mRNA. Second step. Next, the information contained in the mRNA molecule is translated into the language of amino acids, which are the building blocks of proteins. In the third step, this language tells the cell's protein-making machinery the precise order in which to link the amino acids to produce a specific protein. I know this process can seem a little complicated, but think of it as a game of telephone. For example, if I tell my friend the phrase Notre Dame, and they interpret the message and pass it on to the next person, and he interprets it and passes on to the next person, and so on and so forth. Everyone understands the message, just as the body can understand the message, but in different signs and signals. DNA sequences get converted into messages that proteins can use to communicate with the body, so everything is working smoothly. Thank you guys so much for watching my video. I hope you enjoyed it very much.